All right. So this is going to be specifically teaching you about DNA structure. We did learn a little bit earlier in the year, but we're going to go into more detail now. It matches up to Chapter 8, Section 2 in your textbook. So whenever you need to get extra information, that's where you're going to go for that. So we're going to look at the history of how they figured out what DNA is, what does it look like, that kind of stuff. So we're going to start with 1928. So we really haven't known a lot about DNA for very long, only about 100 years, really. So Frederick Griffith was experimenting with the bacteria that cause pneumonia. Pneumonia is a lung disease, and he discovered something. He called it a transforming principle because at the time he didn't know that it was DNA, but he knew that this stuff could be passed from these dead bacteria to live bacteria. In a moment, I'm going to show you a video clip to explain his experiment more in detail. And this picture is kind of a really quick summary of his experiment. So this bacteria that can cause pneumonia, there's a bad version of it, and then there's a version of it that's harmless. So the S one is the bad one, that's deadly, and the R version is just, you know, it doesn't do anything to you. And so he tested different mice. He tried injecting the really bad bacteria into the mice, and of course they died. And then he injected the harmless bacteria into mice, and they were fine. Then he took the really bad bacteria and he heat killed them. That means he heated it up until the bacteria were dead. They were no longer functioning. And then he injected them into mice, and the mice were fine, which is what he expected. But then he took the heat-killed deadly bacteria and mixed it with the harmless bacteria and injected that into the mice, and he found the mice died when they were injected with that. And then when he looked at the mice, at the blood and the bacteria that they had, he noticed that these bacteria were the harmful kind again, even though they were dead initially when he injected them. So he found that to be very interesting, and he thought that something had to pass from those dead, bad bacteria into the live ones. And then in 1944, Oswald Avery did some more experiments, and summarizing really quick, he found that the DNA was the molecule that transmits genetic information from one generation to the next. So this video will kind of summarize both of those gentlemen, what they did.
But in addition to DNA, chromosomes also contain protein, and for the first half of the century, protein stole the show. A protein consists of an irregular sequence of smaller amino acids. There are 20 different amino acids, which can be arranged into any possible combination. As the protein grows longer, the possibilities multiply astronomically. DNA, on the other hand, has an alphabet of only 4 letters, which were thought to be arranged in a monotonous sequence. It's no wonder that proteins seemed a better candidate to carry the language of heredity. It took a couple of scientific surprises for DNA to finally prevail as the stuff of the gene. The first took place in 1928 in the London lab of Frederick Griffith, a physician. He was studying 2 different strains of a bacteria called pneumococcus. One strain had an outer capsule that was highly infectious. When Griffith injected these potent bacteria into a lab mouse, it got sick and died. No surprise there. His next step was to kill these bacteria by heating them. When he injected the dead bacteria into a second mouse, nothing happened. Again, no surprise. Griffith's other strain of pneumococcus lacked the outer capsule of the first and were harmless. When he injected these bacteria into a third mouse, again as expected, they had no effect. Lastly, he combined the second strain with the killed version of the first strain. To his great surprise, this combination proved deadly. When Griffith examined the dead mouse, he found live bacteria that looked just like the infectious strain. This suggested to him that some factor had passed from the dead bacteria into the live ones to transform them. He also discovered that this transformation was maintained in all subsequent generations. So Griffith's transformation factor must have contained genetic material. Across the ocean in New York, a team of scientists at the Rockefeller Institute set out to identify Griffith's transformation factor. They were led by Canadian-born Oswald Avery, who, like Griffith, was a physician interested in the pneumococcal. Avery's team carefully separated out the chemical components in Griffith's dead bacteria and added them, one at a time, to living cultures of his benign bacteria. They found that the transformation factor was pure DNA. It was an astonishing result.
All right. Okay, so introduction to DNA. Since the first half of the 1900s, scientists have known that DNA, or deoxyribonucleic acid, was the molecule responsible for an organism's traits. However, it was not until 1953 that scientists James Watson and Francis Crick first solved the scientific or the specific structure of DNA. The story of the discovery of DNA, of its structure, involves several scientists who were fiercely competitive to solve the shape of DNA. So in this activity, you'll be playing the role of Watson and Crick, trying to solve the structure of DNA using similar kinds of information that the scientists back then used to do the same thing. So here on this slide, you guys are going to be answering these questions. So you're going to take a look at this picture of the DNA. It's been magnified 850,000 times through a scanning electron microscope. So based on the photo, I want you to describe the shape of a DNA molecule, just based on what you see in this picture. What is the shape that you see here? And then letter B is asking, based upon this image again, how many strands or sides or pieces is DNA made of that you can tell from this picture? Okay, so this question is based on your interpretation of this picture. There's no really right or wrong answer. There are many answers in the room that I saw that look similar. I'm going to show you what other possible answers are. You do not have to change your answer unless you really want to. But so an example of how you could describe this shape, I saw a lot of people put twisted as a word, or spiral. Or some people put ropes. So I think I saw someone wrote worms or chain. So those are all descriptive terms that you could use to describe what you see here. And then based on the image, how many strands or sides does it look like DNA is made of? I did see a variety of numbers, but if you look, you can almost pick out two strings, so to speak, here. If you were to watch and follow this shape, it's kind of, you know, S-shaped curves here that it kind of flows through the picture. So it kind of looks like there's two there, but again, if you picked out more than that, that's fine as well. Okay, here's a reality check. The previous image was not possible in the 1950s. The technology for this image was not perfected until decades later. But one scientist, Rosalind Franklin, did develop the image that you see below, which can be interpreted by a trained scientist to draw conclusions from. The image is called an X-ray crystallographic, whereby X-rays can be used to produce images like this one that she produced. So here's Rosalind Franklin. She was working on this stuff in 1952. She used X-ray diffraction to produce photographs that revealed an X-shaped pattern to DNA. And this photo would later be critical to Watson and Crick's discovery. If she had not been able to create this image, they would never have been able to figure out the actual structure to DNA. So this was a very integral part to their discovery. So we're going to watch a video that kind of explains how taking an X-ray of DNA can tell you what its shape looks like.
Almost 100 years later, X-ray crystallography is still used for the same purpose. The molecule complex and the equipment is sophisticated, but the basic principle remains the same. The molecule being studied is converted into a crystal, which locks its atoms into a uniform, three-dimensional structure. X-rays are then used as a crystal. As they pass through, they are bent or diffracted by the clouds of electrons that surround these atoms. These scattered X-rays are recorded on a film or photosensitive receptor as a pattern of dots that is unique to that particular molecule.
We can figure out from this the direction of the scatter or where the electrons were that produced that scattering pattern, and from that, the shape of the molecule.
By the early 1950s, X-ray diffraction of DNA revealed some tantalizing clues about its structure. But the X-ray dots were the tiny peepholes through which only certain features of the DNA molecule were visible. One feature the dots revealed is that the nucleotides in a DNA chain join together to form a backbone of alternating sugars and phosphates. The dots also show that the DNA backbone curls around its axis to form a corkscrew or helix shape. The dots also suggested that a DNA molecule is composed of two such helical backbones winding around each other. The resulting structure is called a double helix. However, the X-ray photos were silent on the third component of the nucleotide, the four different bases. Because of their variability, they scattered X-rays too randomly to form dots. They therefore remained invisible. But the bases had to be within the double helix, attached at right angles to the sugars. Opposing bases must somehow bind to each other, gluing the two backbones together. It had long been known that the four bases come in two different shapes. A and G are larger two-ring structures called purines, and C and T are smaller one-ring structures called pyrimidines. Scientists also knew that identical bases could join together side by side through hydrogen bonding. But such base pairs are either too big or too small to match what they knew was the diameter of the double helix. Also, such base pairs could not have produced the very regular shape suggested by the X-ray photos, which resembled a smooth-walled cylinder. To fit them in, the cylinder would have to bulge out for the larger purines and shrink in for the smaller pyrimidines.
Okay. All right, so now that you know the basic shape of the molecule of DNA, it's time to understand it in greater detail. It has been known for a long time that the nucleic acids, like DNA, they're made up of monomers called nucleotides. And we learned this earlier, way back in chapter 2. There is 6 feet of DNA in every single one of your cells. And all that DNA is made up of billions of only 4 kinds of nucleotides. So you're going to either use your textbook, the virtual textbook, or you can just Google on the internet. I want you to list the 4 different names of DNA nucleotides. They did mention them in the video, too, if you were watching. looks like you guys are good Googlers. So the 4 DNA nucleotides or bases are adenine, thymine, cytosine, and guanine. Those are the 4 that you should have found. Many times they're just shortened and we just use their first letter, ATGC. But it's adenine, thymine, cytosine, guanine. You will need to know them. You will need to memorize them. Make sure you know how to spell them. Okay, so now again, hopefully you still have your Google page open. I want you to look up what the shape or the different parts of a nucleotide are. You're going to click and drag the names of the different parts. We've got hydrogen bonds, nitrogen bases, phosphate group, and deoxyribose sugar. And the structures we're labeling, there's 2 of these green circles, 1 on each side. So the nucleotides are made up of 3 parts each. There's 2 nucleotides here, and the lines in between them represent something holding them together. So I'll give you another minute to Google that or go use your textbook. All right, let's see how you did with these. I'm going to go across the top first. The pink pentagon, it's a 5-carbon sugar that's called deoxyribose. And then these lines are holding the bases together here. Those are going to be the hydrogen bonds. And then the green circle all the way over here to the right, that is a phosphate group. And then, so again, the green circle on the other side is the phosphate group. The double ring shape and the single ring shape here, those are both nitrogen bases. They're 2 different types. And then our pink pentagon is our deoxyribose sugar. So make sure you have those labeled correctly. It looks like when I walked around, you guys all had them pretty accurate. Good. Okay, so this picture is just showing you they fall into 2 categories, these nitrogen bases do. They're either labeled as purines or pyrimidines. The purines have a double ring structure, and that's adenine and guanine. And then the pyrimidines have a single ring structure, and that would be thymine and cytosine. So we're going to use now the little cards on the right, and you're going to click and drag them. Does it describe a purine? Is it describing a pyrimidine? Or does it fall under all kinds of nucleotides, any kind of nucleotide? So we've got adenine, cytosine, deoxyribose, thymine, the phosphate group, guanine, a 2-ring structure, a 1-ring structure. So with the last couple slides that we've completed, and this previous slide too, those should all help you figure out where to put these cards. So you guys are using this slide that we just filled in. There's two different nucleotides here. So the sugar, the phosphate, and this base, the orange double ring one, that's one nucleotide. And then this is another nucleotide over here, okay? They each have a phosphate group, they each have a sugar, and they each have a base. The purines are different than the pyrimidines. Some of them have a double ring, some have a single ring. And looking at the clock, we're running a little short on time. So I'm going to have, hang on, you guys, don't go anywhere. I'm going to have you guys finish this slide for homework. And you need to go back into today's folder and answer the exit ticket question. The exit ticket question is having you design a 3-frame comic strip based on today's lesson. And you're submitting that by tomorrow, okay? Please do not forget to do that.